Hello, everyone. Welcome to the annual Louise M. Slaughter National DNA Day Lecture. This lecture is named in honor of Congresswoman Louise M. Slaughter for many reasons. Among these, in 2003, she led a group of legislators who passed a concurrent resolution creating National DNA Day to celebrate the completion of the Human Genome Project and the 50th anniversary of the discovery of DNA structure, the iconic double helix. Sadly, Representative Slaughter passed away in 2018 after a barrier-breaking career dedicated to science and public service. She was one of the earliest champions for genomics on Capitol Hill and a steadfast advocate for policies that have allowed genomics research to advance to the point where it is today. It is also thanks to her that we have a law called the Genetic Information Non-Discrimination Act, or GINA. This law prohibits genetic discrimination, and its protections allow patients and research participants to undergo genetic and genomic testing without fear that their results will be used to negatively impact their job or access to health insurance. And now it is my honor to introduce today's fantastic speaker, Dr. Carter Clinton. Dr. Clinton is a recent graduate of Howard University, where he was named Graduate Student of the Year, and former assistant curator of the W. Montague Cobb Research Laboratory. He is now a postdoctoral scholar at Pennsylvania State University in the departments of anthropology and biology. Dr. Clinton is also a National Geographic Explorer and Cosmos Foundation Scholar. Initiated during Dr. Clinton's graduate career, his research explores the lifestyles and causes of death of the 17th and 18th century New York African burial ground population, a community of free and enslaved Africans who once lived in present-day Manhattan. Through soil chemistry, human microbiome, and bioethical analyses, Dr. Clinton has developed a protocol to safely and non-destructively investigate historical populations without disturbing their remains. Currently, Dr. Clinton explores the diversity within the African contribution of contemporary African Americans using global data sets and big data analyses. He is working to identify the ancestry-derived contribution of complex diseases like diabetes, asthma, and hypertension. His research interests include studying unidentified historic African descended peoples to fill in the genetic identity gaps created by the transatlantic slave trade. Dr. Clinton's ultimate goals are to compare genomic data of historical African American populations with living populations to address and combat health disparities, better understand disease susceptibilities, and contribute to the necessary underlying research needed to make precision medicine possible and accessible for everyone. His talk today is titled Investigating New York African Burial Ground Population Soil Samples to Better Understand Health Disparities in Living African Americans and the Urgency for Diversity in Genomic Databases, a Microbiome Analysis. Welcome, Dr. Clinton.
Yes, thank you again for that wonderful introduction. I am excited to be here. It has been a long time coming, but the day is finally here. One thing I like to note, I know that the title is extremely long, but we'll be talking about all of these wonderful things wrapped up in one nice bow. Majority of the work that I will go through today, much of the results, was generated at Howard University during my time as a graduate student. I'd also like to mention that I'm honored to be chosen to give the 2022 Louise M. Slaughter National DNA Day Lecture to share my research, speak about the possibilities for positive change in scientific research, for which Congresswoman Slaughter has already initiated the groundwork. And so looking at the past, so we have to start all the way back to the beginning. So I'm showing here the transatlantic slave trade specifically for African Americans, for the trafficking of African Americans from, or continental Africans from continental Africa to the Americas. These are facts that we know very much about. there was an estimated 9 to 12 million Africans trafficked to the New World. They came over by the British, the Portuguese, the Dutch, the French, among others. And many of them were taken from West Central Africa, the Gold Coast, West Africa, again, and as well as many other places. But while we're familiar with these facts, or while we're familiar with the presence or the existence of slavery in the South, we're still mystified about the existence of slavery in the North. However, we have this excellent resource that is the New York African Burial Ground. Not only does it tell us about the institution of slavery in the North, but specifically in New York City. Now, what you're looking at the top of the slide is one map of the New York African Burial Ground, where it's located today, where the maps indicate in 1776. And on the right, we see where the New York African Burial Ground, now a national monument, is located today. What I really like to point out are these landscape factors. So on the left, which we do not see today, is something that we call a freshwater pond, or the collect pond. This served as a major water source for the community in New York at that time. And I'll reference this later on in the talk, so it's a bit important. Another component of this map that I really want to point out is the most famous street in the world, Broadway. So the burial ground is located, if you are familiar with New York, right at the intersection of this famous street, Broadway, and Chambers, right in downtown, Lower Manhattan. Whenever I speak about the New York African Burial Ground, and I guess have to indicate that I go back to the beginning, I really have to go back and say, yes, slavery existed in the North. Yes, slavery existed in New York City. So something that we may not know, a lesser known fact, by 1703, New York actually had more than 42% of its household held enslaved Africans. Now that was second only to the major slave trading port of Charleston, South Carolina. And by 1861, New York threatened to secede from the Union with the South. Why? Because they were profiting so handsomely from the institution of slavery. Now, what exactly is the New York African Burial Ground? Well, it is the largest burial site of free and enslaved Africans ever discovered. It existed during the 17th and 18th centuries. During an unplanned construction project, it was rediscovered in 1991. From there, 400 skeletons were excavated, brought to Howard University, where reports were generated on the skeletal biology, the archaeology, and the history of these remains. It is important to note that these skeletal remains were reburied out of respect for the population and for the descendant community. And so when I came along, I had the pleasure of working with the burial soil from which these skeletal remains were collected with. It is now recognized as a national monument. So this is how it stands today, but the origins of the New York African Burial Ground are very significant. So we have this hypothesis that there were 11 half-free slaves, and they were half-free because they were originally enslaved by the colony, the New Amsterdam colony, or present-day New York, petitioned for their freedom, were granted that freedom, but their offspring would still be enslaved and belong to the colony. So they were half free. Upon their half freedom, they were allotted a plot of land just outside of the New Amsterdam colony. Why? To serve as a barrier against invaders. And it was here that this community, or that these 11 individuals, constructed this burgeoning community. I'll direct your attention to the map on the left-hand side of the screen. So this red line would be the borderline or the Palisade line to the New Amsterdam colony, which later became New York City. And here is where we have the Negro frontier just outside of that border. Now, it is, again, adjacent to the Collect Pond, which is this source of water that they used, but also in thinking that this is a new community, they did not have a means for carrying their loved ones once they passed away into the colony, or even the money to pay to break ground and bury them. They appropriated a piece of land just outside of the border of that colony. As you can see on this historical map, it is named the Negroes Burial Ground, known today as our New York African Burial Ground. So I came along, and being from Brooklyn, being African American, and wanting to know more about this population, and decided to do some research. I really wanted to know, how can I combine this leading edge technology, our new next generation sequencing technology? Specifically for this project, we used 16S rRNA sequencing to explore really anything we could to learn more about this population. The institution of slavery in New York, any evidence of West African retention of burial practices or traditions, identities of these individuals, and even any environmental impacts that we could find by exploring these 400-year-old soil samples. And the way that we did this was, one, we had to figure out a protocol to extract DNA, genomic DNA, to look at all of the bacteria in a sample without having a model for this project. So a question that I most generally get is, well, if you have burial soil, you know it came from an individual, why wouldn't you look for human DNA? Well, because this hadn't been done before, the soil had been buried for 400 years. And I knew that it was a little risky looking for human DNA given the resources that we had. But you were always going to find bacteria wherever you go, especially in soil. So I extracted all genomic DNA from the burial samples. We sequenced these isolates for the 16S gene, which really says whatever is here in this genomic DNA, we only want to look at the bacteria. And once we had all of our bacterial sequences, we made sense of it using this bioinformatic program called Mother. And so I guess the next question to ask is, what are we going to see? Or what do we expect to see? And in looking at simply bacterial DNA, we would expect to see something called the human microbiome, or the necrobiome. So what is the microbiome? Well, all of the bacteria that live within us and even on our bodies that help us to digest foods, to fight off infections, that help to protect us in general, help us with our diets, they also help to decompose our body after death. And what we're finding is that we're still able to detect these bacteria even after we have died, are buried, and have been skeletonized. This picture on the right-hand side, is each of our burial soil samples labeled, and I'll go into more detail in a second. So more about that microbiome. I think that it is a hot topic right now. It is what's becoming our commercial trend right now. We have companies that are coming across our screens saying, hey, send in your saliva, we'll tell you, why your breath stinks. Send in your poop, we'll tell you if you have cancer, right? It is, again, really just this hot topic. We have all of these kits that are popping up that provide this service for us, that tell us what's there, what these microbes are doing, if they're helping us to break down complex carbohydrates, or if they're maybe making us sick. right? We even have this incorporation of protecting our microbiome in the soap that we use. And we even have this interest to the extent that we're extending this research to our four-legged loved ones. So we even have microbiome kits for our pets to make sure that we are giving them the best diets so they can live long and healthy lives. Now, why should we study the human microbiome? And not that this was an off choice from our human ancient DNA analysis, but in reality, the human body has 10 times more microbial cells than we do our own cells. So looking at it sheerly from the numbers, the human genome has around 20,000 coding genes, right? Our human microbiome has around 3.3 million genes. That means that in the age of data analysis, that is way more information to be able to draw conclusions upon. And the microbiome has also been called so informative that it's been titled our second genome, and for reasons that I will get into in a second. Some clinicians even hypothesize that around 90% of all diseases can be traced back in some way, shape, or form to the gut and to microbiome health. Now, we wanted to take all of this information, or this powerfully informative application, and we came up with some objectives. So we had 69 samples that corresponded to 54 burials. So in each one of those jars, in the previous picture, we had, for example, one burial with multiple samples. So for example, burial 10 with cranial soil, pelvic soil, and say tibial soil. So we know exactly where these soil samples are coming from. We wanted to identify the differences between the New York African burial ground samples and non-burial samples. What does that microbiome profile look like? And can we see evidence of any infectious diseases associated with any of these burials? To do this, we needed a comparison. So we got our control samples from select locations around Lower Manhattan. Now, if you have been to New York, you are very well aware that it is the concrete jungle. There are not a lot of green spaces, and even the ones that are available or that do exist are not available. You do not have access to them. So we retrieved our controls as an opportunistic sampling process based on proximity, availability, and accessibility. Now, I know that, again, this talk spans many different disciplines. So for those of you who are not from that science background, I'm going to take you back to high school biology for a second. So we're going to be talking about our taxonomy, or the breakdowns for each of these bacteria, specifically the phylum, this taxonomic level, and our species level. And the most familiar thing that I can associate to you, right? If we are Homo sapiens, sapiens is our species. It is the most specific classifications for us as individuals. So for our 16S rRNA gene sequencing, we wanted to see everything that was in a sample. Again, because this had not been done before, we wanted to explore every aspect of the research that we could. We broke this down by relative abundance in each of the soil samples. So here we have combined the New York African burial ground samples and also our control samples. We've listed them I've listed them from greatest to least, with Firmicutes being the most abundant and Fusobacteria being the least abundant. And so in simpler terms, I wanted to break down what these differences were, yes, between burial and non-burial samples, but my even bigger question was, would I see something significant and would I have a dissertation project? So in looking at this comparison, so everything in red is our New York African burial ground samples. Everything in blue is going to be our control samples. And so we saw significant differences at several different taxonomic levels, but again, we are just showing the phylum level for now. The two most striking differences that we observed, we saw increased abundance in our New York African burial ground samples for our Firmicutes, and we saw an increased abundance in our control samples for our Acetobacteria. And so this made me very happy that we see a difference somewhere, but also I wanted to take a closer look and see what those differences were and what the driving factors were that were responsible for these differences. And so what I did then was I took a median representative from each of the groups. So we have burial 150 representing our New York African burial ground samples or our burial samples. And we have City Hall Park, which is just steps away from the New York African burial ground monument, representing our control samples. And so again, if I can bring your attention to that Firmicutes contribution, in our burial samples versus that very limited contribution in our control samples. So this led me to ask another question. Well, what specific bacteria do we see that are contributing in this area? We see things like Lactobacillus, Ruminococcus, Clostridium, things that we commonly see in the gut bacteria of living people, regardless of age, diet, or population. We don't see that representation here in our control samples, but what we did see in that increased abundance in our Acetobacteria was newly identified unnamed microbial taxa. Why? Because this is soil bacteria. It is rich in bacteria, many of which we're only now being able to detect and identify with the advent of next generation sequencing. Before, we could only detect and identify the things that we could culture in the lab. And so we're seeing many more unnamed taxa. And so my next question was, okay, well, those are stark differences as far as abundance, but what about these levels where they are comparable, where they almost look the same? And so I explored the portions of proteobacteria. And so in our burial samples, not just, our representative burial 150, but in all of our burial samples, we see things like Kingella, Salmonella, Burkholderia, again, that we commonly see in the human gut microbiome. And on the other hand, for our controls or our non-burial samples, we see things like Pelamonas, Azotobacter, things that are responsible for the pH in soil and specifically that are native to the wetland soil environment. Another thing that we noticed as we compared each of the burials to each other is that no one looked like another. And so my hypothesis here is that we can use this for forensic purposes, for means of individual identity, to draw inferences about who the person was. For example, in that forensic case, if we are looking for a person who was a smoker, we know the body was here, it just happens so that it is not here at this moment. Does that residual evidence represent the microbiome profile of a smoker? Well, maybe this one doesn't. However, from what we're finding, this person seemed to eat a lot of dairy, right? In addition, what we're finding is that it may be possible that we're seeing population and ancestral origins or distinctions between populations due to environment and diet. And so the ability to use this when human remains are unavailable, or you just do not have access to those things, or you do not want to disturb those remains, gives us the ability to work backwards for these forensic purposes. And so now I really just want to orient you to the outline of the burial ground. So everything in this rectangle, where it is represented on our map. So we have our street view that we used earlier. That rectangle that I just outlined is the same thing as this rectangular excavation site from the early 90s for the New York African Burial Ground Project. And here is our reference for, again, that most famous street in the world, Broadway. So in looking at the geographic distribution of these burials and what we found, but specifically for disease pathogen, we found evidence, which I think is really fascinating, evidence of these different or possible pathogenic bacteria in several burials. It is important to note here that our analysis only distinguished down to the genus level. And so future analyses to decipher species and strain is going to be necessary to really confirm these conclusions. But some of the more interesting ones, Shigella, for example, from dysentery, which we know ran rampant on slave ships coming to the US. But these aren't the only ones that we found. Throughout the burial, we saw things that were indicators for cholera, or even indicators for meningitis. So we're not only able to see the microbiome profile of the individual, but possibly able to attach a cause of death. So for these bacterial DNA conclusions, so one, our soil samples show the New York African burial ground looks very similar to living human gut microbiome diversity and communities. The identification of this human microbiome profile can act as a form of identification, possibly an indicator for diet, identification of infectious diseases, which then may give us some insight about the occupation of this person. Did they work at the ports? Did they work indoors? Or anything about their lived experience. We are constructing this protocol that is a non-destructive one to investigate historic burial grounds or historic populations across the country. And that way we do not need to destroy or disturb human remains or human skeletons, especially when that local descendant community, that is their request to not do so. And finally, and perhaps more importantly, circling back around to that initial question. Now that we are seeing this definitive evidence of the human via human microbiome, can we now move forward and look for human DNA, which is part of the main reason why I came to Penn State for this analysis. Now, I told you why the human microbiome was informative, why it would be nice to study in lieu of the human ancient DNA or looking for human ancient DNA. Well, we have several different factors that are going to tell us different things about different people, but also different things about ourselves as far as the health of our bodies, but also our brain health. I'll get into that in a second. So we have the presence of different bacteria at different locations of our body. So our our armpits, our feet, the reason why, some people's feet may smell like corn chips, all thanks to that microbiome, that bacteria in that area. In other examples, the microbiome has been linked to diseases or disorders like anxiety, schizophrenia, colon cancer, among others. And I'll get into the specifics on the next slide. But there are many other things that affect the microbiome. It's not just the microbiome and then the genetics. It's just like everything else in this world, they're always interacting and affecting each other. So we have our immune systems, our BMI, our diet and our lifestyle, our evolutionary history, which we will get into, the use of antibiotics, our age, and so many other interactions. Now, more specifically, we spoke about depression. What we see is that more than 1/3 of depression sufferers have what we call a leaky gut. Colon cancer individuals, we have sugar-loving microbes that fuel the presence of that colon cancer. Obesity and diabetes, we have a dysbiosis. That means that it is very likely that the bacteria in which are common to everybody are just very different in the proportions in which they show up. Rheumatoid arthritis, we may have low levels of good bacteria and high levels of bad bacteria. Parkinson's disease and autism, people suffering from these diseases have a very different bacterial community than do healthy people. And again, we all have, there's no one microbiome profile, but there are similarities based on our environments, our diets, and who we are as individuals. And then finally, I wanted to end off with asthma. So we have indicators from our human microbiome, which is microbial dysbiosis, but we're also looking at an airway microbiome. So you're getting this evidence from two different places, again, that need to work together. The microbial dysbiosis, fewer of those things that we said is most common in the guts of living individuals, lactobacillus, bacteroides, etc. And in our lungs or airway microbiomes, the presence of these species that create this worse lung function. Now, I kind of want to use asthma for a second. We know that that African Americans have higher asthma-related morbidity and mortality rates compared to European Americans. Also, cases of prevalence is given by the examples on our screen, okay? The causes of asthma are not just genetic or due to an imbalance in the microbiome. It's more than that. It's other interactions. We see things that are biological or even beyond our biological, from our genetics, our nutrition, our microbiome, and our diet, to our behavioral and our socio-cultural backgrounds with socioeconomic status. Do you have health insurance, psychosocial stressors, and even, again, our environment. What does your neighborhood look like? The level of pollution, our housing conditions. Do you have access to healthcare? All of these things affect the prevalence of asthma, disproportionately affecting African Americans. While we think that we have the genetics all wrapped up in a nice little bow, we know that all of these things are affecting the prevalence, the morbidity rates of asthma. We don't have it all figured out. So another likely explanation that leads to this disparity is that, well, when the genes that confer risk may be the same across ethnic groups. And I want to point your attention here. So really, this is just an admixture mapping analysis. This is, again, the work that I am now moving into at Penn State with contemporary or living African Americans. We're looking at the global ancestry of individuals, specifically of African Americans, and then local ancestry, or where does this ancestry show up in these chromosome blocks, okay? And when we look at those chromosome blocks, we associate these areas of interest with these markers that are indicators for this trait of interest. In our case today, asthma. So it may be the same across ethnic groups. However, the frequency of the polymorphisms associated with asthma may be different according to ancestry. So there are several different examples of variation in frequencies of high risk in candidate genes associated with asthma. So it's not just about the fact that they're there, that we are observing the presence of these variants, but the frequencies of these variants. Finally, the number of association studies exploring asthma and associated genetic markers are overwhelmingly performed on European Americans, although it is African Americans who are disproportionately affected. So the causes of asthma are not just genetic, again, not just due to an imbalance in the microbiome or the environment or socioeconomic factors. It is the combination of all of these things. These are all things that underlie some of the disparities in the health burden of asthma, specifically in the US. So this is where our systems biology or interactions biology comes into play. It's a biological issue, an environmental issue, a healthcare issue, but there is one other factor to take into account. All of this is compounded by inaccurate measures of lung capacity in African Americans. So going back to the historical origins of the spirometer. So we have this guy, Samuel Cartwright. He was a Southern physician. He was the first to use a spirometer and look at these measurements and how they differed, the measurements, of lung capacity and how they differed between enslaved Africans and white counterparts. But remember, this was also the guy who came up with drapetomania. Drapetomania was classified as a disease for runaway slaves, in that, plantation owners provided this comfortable setting, and for enslaved Africans to want to run away, something must have been wrong with them. And although this idea was debunked, it was pseudoscience, and it was also shown to be part of the edifice of scientific racism, is still used worldwide today. So we have something called a race correction factor. And in many cases, clinicians today who still use this spirometer to look at the lung capacity of certain individuals have to press a button. They don't know what this button does, but it is the race correction factor. It is already built in to this analysis. So if you are a member of a population other than European descent, then you would automatically have this lower indicator of lung capacity. Now, this is not the only example that we've had throughout history that have affected our healthcare system and our science. So here are some great examples of non-scientific or science adjacent literature that really explain the long-term effects of mistreatment for African Americans and how it contributes to biased conclusions and negatively affects African Americans in medicine. They really explain, how leading edge technology can be used to control the past and change the narrative, how this social construct of race is a determiner of biology, the long-term effects of slavery, and how that has implications for today in healthcare and in scientific research. We don't just see this, the thing that really gets me, we can make it to Mars, but we are still kind of stuck in many ways in this area. And we're still even though we have some of these acknowledgements, we are still producing this science that continues and perpetuates these thoughts and these notions. So we have papers that are coming out as late as 2019. Do your genes influence how much money you earn? Okay, without thinking or taking into account, whether some people were born with a trust fund or not, right? How about the fact that this paper came out 2012, genes influence criminal behavior. So this paper says that antisocial children exhibit greater resistance to authority as they grow older. We have this 2018 paper, Years of Education Influenced by Genetic Makeup, Enormous Study Finds. So again, we have this study that reports on educational attainment, again, not taking into account socioeconomic status, how much access to scholarships we have, how certain students are able to pay for tuition versus other students who need to take out these student loans to be able to go to school, right? And that student loan conversation is a whole nother lecture in itself. And finally, we have papers that, have come out as late as last year, 2021, ancient DNA researchers set ethics guidelines for their work, where really we have these researchers saying, yes, we need to look for ancient DNA. We need to invade these populations, these underserved populations, because we're doing you a favor, because it'll benefit us for science. This is an issue. I won't go beyond what the title says, that it has created, has drawn criticism from others, but we need to be rethinking the way that we are doing the science in certain areas. Now, I think what they were trying to get to is that we don't have this representation. And so we have to go to these underrepresented populations to enrich the global databases. And in that is very true. Our global databases, the way that it stands, well, at least as late as 2019, this is came from a paper from Sarah Tishkoff and her group, okay? We see that the global genomic databases are overwhelmingly comprised of European individuals. So my question, we have Africans listed as 2%, right? So let me start. It's not just about acknowledging that something needs to be changed in the way that we need to approach health disparities. Even if we had an even playing field, meaning quality of environment, neighborhood, pollution, access to healthcare, and other factors, we'd still be limited by the lack of representation in databases. So populations less frequently represented in genomic databases equals populations that we know very little about. So we need to enrich, we need to do it correctly and ethically, but we need to enrich our databases, because if for no other reason, representation provides a resource for physicians to make more informed health-related decisions to address health disparities in medicine. And another point of concern, so seeing the value of studying people of African descent, let's think about it like this. If all humankind originated in Africa, shouldn't the percentages be flipped? If all populations carry a subset of African diversity, shouldn't we be studying African descended people so that we can learn more about them, all populations, and even the origins of our species? Without the full picture, we can never understand the full scope of what's going on. And I would be remiss to not include one of these excellent depictions of how important diversity is by, coming out of this excellent NHGRI communications team. So genomics research only based on people with European ancestry? I don't think so. Genomic research based on every ancestral group's DNA is the future, is the way we need to go. So conclusions, I know I'm coming up on time. The goal. So I believe that really the overlap between historical resources, the study of these historical populations like the New York African burial ground with biological science and the combination of data science is really the sweet spot that will transform human health. So we need a combination of all of these things, including AI and all of the now emerging algorithms and capabilities to really, the first step is really understanding what we're dealing with in order to prepare for a better health outcome for everybody. So the overall conclusions: new approaches to address health disparities in scientific research. Use what we understand, use what we're learning by looking to the past to reframe our thinking about science and medicine. Go back to the foundation. Acknowledge the biases in scientific research. Consider that precision medicine may also need to be combined with precision nutrition, but also other precision omics to create that full scope, that full picture. We really should enforce legislation to protect burial sites of historic populations for research for this reason, or not, based on the desires and the demands of the descendant community. And finally, the key is that diversifying our genomic databases is the key to our growth as researchers, as people, as a community, and essentially as a population. And so with that, I would like to say thank you. Thank you for your time. Thank you to NHGRI for having me.
Thank you very much, Dr. Quinton. I'll give a round of applause for those that are in the audience. And thank you very much for the talk, going from the smallest of things to the biggest of societal issues. And so we have about 10, 12 minutes to go through some of the questions that have come in and some discussion time for everyone to listen to. I just want to, I'll start with some of the questions that have come in from the audience. I do have a few questions myself, but I want to get to a couple that are somewhat key and that Cynthia Williams wanted to know are the methods that you're using being disseminated in a way so they could be used elsewhere in the country? And my guess is it's easy to point folks to papers that I saw. One was under review. And so is there an obvious place to help find the methods? It looks like you've combined microbiome, ancient DNA, soil sampling, environmental and archaeology. So
Yes.
Do you want to comment on how other people could do this in their own research backyard?
Yes, absolutely. I think one, that's the goal, to be able to create this non-destructive protocol and for it to be used. Right now, we have not gotten that publication out for others to use to apply it to other burial grounds, but that is the absolute goal. I think even in previous talks and through other publications, people have reached out. So we are already implementing this protocol at other sites in North Carolina, in Maine, and in Pennsylvania. So shortly, you should see, publicly available this protocol that can be extended to other burial grounds.
So Cynthia Williams and others, be sure to search for Carter Clinton's publications as they came up going forward, and you'll be able to get the protocol. Along the lines that someone also, Sarah Bowden, asked if how much dirt, I'm not sure I'm going to look at soil and dirt the same way ever again after this talk, but how much do you need, and I'm sure this will be in the paper, and whether or not if you do samples from the same spot, how much variation you see. Remarkably, I think you talked about being able to sample, if you know the orientation in which the person was interned, being able to sample on different parts of their body, the dirt under different parts of their body.
Yes, that's correct. So with the New York African Burial Ground, we were a bit limited in that the soil samples were collected with the skeletal remains at the time of excavation, before we really had a grasp on this technology, any DNA technology and next generation sequencing even. So I don't come along until 2015, and we now have the technology, we don't have the remains, but we have the soil. And so that collection process was really reflective of what was important to the archaeologists in reference to the skeletal remains. Now, when we go out and we do this, well, there's a huge difference between known burials and unknown burials, because unknown burials, you really don't know exactly where they are, how deep, but we have, this is also going to come out in the publication as well, we have a protocol where we take specific samples from each part of the body, kind of following the way that they did at the original excavation, to see those differences, to see that variation. And we do see significant variation between different sites of the body, even after, hundreds of years after the body has been decomposed.
And there's a lot of interest, I'll tell you, in the methods. I'm not going to go through each of the questions, but everyone's really anxious to be able to look at the methods. Someone hasn't asked yet. We'll stay with the technical for a bit, then we'll go with the bigger picture societal things in a second. But I imagine someone would ask, could this be used as a core sampling kind of method to figure out whether there were burials, if you couldn't find out any other way, or you had no way of doing the destructive digging? Could cores be driven through?
I think absolutely so. However, with the core, you don't want to mistakenly hit a body if there are skeletal remains there. So, I mean, with the projects that I'm involved in, they do ground penetrating radar to see if anything is there. Does it look like a casket or a body? I would say to use some type of detecting mechanism to or even go back to historical maps, any way you can, to see if there are bodies there, how they are stratigraphically arranged. But absolutely, you can use a core method, whatever you find, based on exactly what I just spoke about. Does it more closely like burial soil or even gut microbiomes that we see in the scientific literature, or just simply environmental microbiomes, environmental bacteria?
We're going to zoom out a bit, just to say that people will be looking for your methods. So just with that, zoom out more to some of the societal.
It's coming soon. I graduated last year in the pandemic, so it has been a wild ride, but it is coming.
So now we'll zoom out a little bit into more of the social issues, and I'll get to a couple of the really good questions in the chat. But first, I just want to ask you, and this may have happened before you came on the scene for the New York burial projects, do you know what kind of community involvement and outreach that might have been done? And this ties into some of the questions we're getting from the audience as well.
Yeah. Yes, absolutely. So I think that the New York African Burial Ground, the excavation process, the archaeological process, the bioanthropological steps that they used in the early 90s is a really great example of community-based research. So upon discovery of the burial ground, it was really the local descendant community in New York that halted the construction of that federal building, because initially the burial ground was discovered when they broke ground, but they continued to build that building, and it was the local community who came in, grassroots organizers, and it wasn't until later that government officials and even David Dinkins became involved. But it really was controlled, organized, and led by the descendant community. And the former director of the Manu Cobb Research Lab, Michael Blakey, was the director of that project. And so he, coming from Howard, went to the community and really let them lay out what they wanted to find, the research that they wanted done, what was too destructive, what are the limits, what do you want to learn about the history and the relatedness to this population. And I think that, I'll go back to the beginning, this is a great example of how many other projects should be performed. If you are exploring the historical biology of a historical population, you need to go to that descendant population, especially if you are outside of that population. But you also have to consider the implications and your privileges, even if you are from that population. So I could say, and I just mentioned this at a previous talk, I could say because I'm African American and I'm from New York, I'm the perfect person to do this research, but that really couldn't be farther from the truth. We always have to, as researchers, interrogate our power and our privilege, and acknowledge the fact that we're serving this local community.
And I think that's a good message to the many people who want to start to deploy this method, is to, one of the first places to start is with the community involvement.
Yeah.
Before you start digging,
Yes.
Definitely before you start digging. It also brings up, Reba McDaniel pointed out something that we know and we need to do much better at, and that's rebuild the trust of the African community to participate in science. And actually our institute and others are trying to make sure that not just participate or be participants, but to be the researchers, as you
Yes.
then that actually goes miles. It gives an outlet for people to do new things, and to become scientists themselves, which we really encourage. I'll go back to the really very intriguing, can you get cause of death, or can you get afflictions? That's something from the soil, that something may have happened several hundred years ago. And Trisha Chakrabarty asked, and I think you were suggesting that the people who died and were buried were more susceptible or even had those diseases, and that's what your data is somewhat showing. But she wondered whether there's some of the diseases, and maybe she was talking about asthma, that were maybe less prevalent 200 years ago. I'm not sure I know the answer if they were. So there's a difference between finding the signature of an infectious disease in the soil of someone who may have died from that infectious disease or lived with it, versus finding a complex disease like asthma. Is there a way to think about that from your data?
I think that's a great question. I personally, again, want to answer all of the questions that we can answer. For now, what we have definitively, really seen is that if we're looking for bacterial DNA, we are seeing the evidence of these infectious diseases. So the person either, had it at one point, or it was responsible for their death. But in looking at microbiome proportions or contributions for complex diseases, I think it will take more research. It will take future research and comparisons. And again, building that reference, increasing that diversity, so we have something to compare it to really draw those conclusions. I will not say that we can, at least not with the resources that I have been exposed to, do that at this point in time. But I think there will be a point on the horizon where we will be able to do things like that.
I think this feeds into another point that Reba McDaniel and also Zoe Diamond made. Reba asked whether we could understand diets of people, looking at the microbiome that surrounded their burial. And Zoe was, again, thinking about infectious disease sleuthing and how we could make conclusions about that. So a lot more to come. I want to just end, because I know we have just a minute left, with a question that Isabel May posted and asking about the role of humanities-based scholarship. History, cultural studies have probably put certain parts of anthropology and social science as well. how do they fit into the work? And I think this comes from the question comes from the point of someone who would like to teach some of what you're doing. How do educators teach the approach you're taking in the undergraduate and graduate programs? And she said it was a wonderful presentation too.
Thank you.
So any tips for folks? It really makes the science come alive. If you're teaching a section on microbiology or genomics, and then you can wrap it up in the kind of story that you're doing. So what's your last final bit words? What's your tips to keep this stuff, make it alive for undergraduates?
Absolutely. I will keep it short, but I will also mention that I answered a similar question to this on our Twitter Q&A about incorporating history or the humanities, anthropology into the genomic research, into the scientific research. And I think that you really can't have one without the other. Even if you're working in a lab and you're working with a cell, the context to which the results that you're finding are only relevant to what has contributed to that cell becoming that cell or being that way or ending up in your lab, for example. So I really think that is a misconnection that I hope that we can resolve, that we can establish in the near future, because it's a two-way street for historians or for people in the humanities. You have these oral histories, you have the historical records, and then you have this scientific evidence that really gives validity or gives weight to all of those stories and all of the things that you have been finding. So I would encourage this collaboration between those in the humanities and those in the sciences moving forward.
Thank you. And on behalf of NHGRI and all those who attended, I thank you for giving today's Louise Slaughter lecture, and happy DNA Day. For those of you that have asked, we will post the recording of this lecture on genome.gov. I'm guessing it usually takes us a week or two to get everything up. And we will also include a link to the Twitter, if we're allowed to, the Twitter conversation that Dr. Clinton had earlier or last week that he just referred to. So if we can send the link to everyone who registered once it's posted, that's probably the easiest way. And if not, I would just suggest you come back to genome.gov the end of next week, and you can find the recording. So thank you very much, and thank you all for your attention.